In bacterium E. coli, this is the inner cytoplasmic membrane and this is the outer membrane. The outer membrane of E. coli has a protein called as PhoRin. This is the cytoplasm. This is the periplasmic space. The inner cytoplasmic membrane separates the cytoplasm from the periplasmic space. The inner cytoplasmic membrane has a transmembrane protein called as PhoR sensor protein. It is present in an inactive form. A protein kinase transmitter domain is present in the cytosolic region of PhoR. This protein kinase transmitter domain has a conserved histidine denoted as capital H. The PhoR sensor protein also has a periplasmic domain. The periplasmic domain is bound to a phosphate. Whenever the phosphate concentration in the periplasmic space decreases or in the environment the phosphate concentration decreases, the periplasmic domain bound phosphate dissociates. As a result, the conformation of PhoR sensor protein changes reversibly. Due to this conformational change, the protein kinase transmitter domain of PhoR sensor protein becomes active. This is the cytoplasmic ATP. This ATP has a gamma phosphate. The activated PhoR sensor protein then transfers the gamma phosphate in ATP to conserved histidine in its active protein kinase transmitter domain. In the cytoplasm of E. coli, a PhoB response regulator protein is present. It is a transcriptional activator. This PhoB response regulator protein has a receiver domain. The receiver domain has an aspartic acid denoted as capital D. Initially, the PhoB response regulator protein is present in its inactive form. The phosphate bound to the conserved histidine of active protein kinase transmitter domain of PhoR sensor protein is then transferred to a specific aspartic acid present in the receiver domain of inactive PhoB response regulator protein. Due to this phosphate transfer, the conformation of PhoB response regulator protein changes. Due to this conformational change, the inactive PhoB response regulator protein becomes active. The active PhoB response regulator protein then activates the transcription of PhoE gene. It also activates the transcription of UGPB gene. It also activates the transcription of PhoA gene. It also activates the transcription of PhoS gene. The proteins produced from these genes then help the cell to respond to low phosphate concentration. Please like, subscribe and share.